Okay, hello everyone. We're going to go through the topic 4 of the GCSE EdXL biology today. This is a triple version, but obviously the combined is all of this stuff minus the stuff that's in red, which we'll make sure is clear as we go through it. So, first topic is natural selection, which I'm sure you've done before. We're just going to add a little bit more to it at this stage. First thing to think about is what is evolution. Natural selection and evolution are not the same thing, but kind of one is a driver of the other. Natural selection causes evolution to happen. So evolution is simply when a species changes over time. It gets bigger, it gets smaller, it gets faster, it gets brighter colors. It doesn't have to be huge dramatic changes, like it can suddenly fly or it can suddenly swim underwater or it can solve problems. The two scientists who led the charge with natural selection and therefore evolution were Charles Darwin and Alfred Russel Wallace, who were both both explorers, I suppose, as much as they were scientists, and they traveled the world looking at animals and plant species, and they made some observations, which were that even within a population or a species, you get variation, and that every species will produce more offspring than the environment can support, with the acceptance that not all of the offspring will survive, but by making lots and lots of offspring, enough will get into adulthood and can then have their own children in the future. We're not going to worry about this, but here, so evolution is obviously a relatively old theory. It's about 200 years old now, hasn't changed a great deal in that time, but it is still relevant today. We use it to classify organisms because we can use common ancestors to work out where things should sit and how related two things are. Common ancestor is obviously just an ancestor that two different species share. It's also used in antibiotic resistance, which is a huge, potentially bigger than COVID deal, and it allows us to understand where antibiotic resistance is coming from, how it happens, and therefore how to prevent it. And finally, it can be used in conservation because we can use genetic analysis to make sure that breeding programs are using the correct individuals. We want breeding programs to use individuals who are the same species but have as much variation as possible, because that will produce yet more variation, and that means a varied population which can survive climate change and viruses and pathogens, that sort of thing. So natural selection. What is natural selection? It's basically survival of the fittest, and we'll look at some examples of it as we go through this section. So a species initially is a group of similar organisms, which I'm sure we would all kind of understand that tigers are all pretty similar to one another, lions are all pretty similar to one another. But we need to go a little bit further than that at GCSE and say that they can interbreed to make fertile offspring. In other words, two tigers can make tigers, and those tigers can have more tigers, in the way that a tiger and a lion can have a ligon, which is very exciting, but your two ligons won't be able to produce more ligons. They are at the end of a road, they're not a continuation. So species, group of similar organisms, but crucially they can produce more of themselves. They are fertile. Are members of the same species identical to one another? No, they're not. You can look around a classroom and see that. You can look at people walking their dogs and see that. They show this thing called variation, which is differences within members of the same species. First example of natural selection is the peppered moth. It's a real example. It's pretty clear what's going on here. So if we look at the image on the right, so where it says left, we have three moths. 1, 2, 3 is a light form, 2 light forms, and there's 1 of the dark form. The first question is just common sense, but it's also the reality of the life of a peppered moth. These are all the same species, I should say. There's variants of the same species. The light form is camouflaged, therefore it's more likely to survive if it's not been eaten. Why do they become more common over time? It's because these ones carry the light gene or the light allele, and when they reproduce and produce more moths, they're more likely to pass on the light form. You've done dominant or recessive right now, which is why the dark form doesn't disappear, but the light form will certainly be more common because it's producing more offspring, and those offspring are more likely to be light-colored than dark-colored because of genetics. When would the dark form become more common? It would be if the climate changed so the bark was black, which actually did happen historically when we produced lots more pollution than we do today, and all of the trees in southern Scotland and England were covered in soot and dirt and dust, and the dark form did indeed become more common, but it's kind of switched around again as we reduce our pollution levels. Second example is peacocks. There are some stunning drawings of some peacocks. We are to put the more variability to attract females, so the one that could best attract a female would be peacock 2, because it has the best display. The whole point of male peacocks having these feathers is simply so they can attract a female peacock. Female peacocks are just camouflage, they don't need to do this sort of stuff. This one has the second best tail, so it would be the second most attractive. This one has the worst tail, so it would be the least attractive. Having said that, everything is a trade-off in science, so although this one is the best able to attract females, it would also struggle to avoid predators. It's got this huge tail to carry around and try and hide. It's going to be harder when you have all of these feathers, so it is a trade-off. So in an environment with lots of predators, which one would become more common? 3 would become more common because it's able to hide from predators. The predators, being lots of them, mean that that's the biggest issue facing them. So these 2 would get eaten, this one would be fine. If you think this one would be the most common, I can live with that, but it would struggle if there were lots and lots of predators. If the predators all become extinct, how would this affect the peacock population variety? If the predators all went extinct for some reason, then over time 2 would become the most common, assuming it hadn't gone extinct, but 2 would become the most common again. And that just shows that natural selection isn't aiming towards this perfect peacock, it's just what is best in each generation. That can come and go depending on the environment. And then a nice summary of the stage of natural selection. If you're asked to summarize natural selection for 4 marks or 6 marks, these are the things you should be talking about. Pictures aren't too important, but they're there to kind of highlight. So we've talked about these things already. So first one and second one kind of go hand in hand, but we think of them as stage 1 and stage 2, which is that any population, any species will overproduce. So there's always more offspring than the environment can support. If you've ever had like mice or rats, they produce hundreds of babies every year. That there's no expectation they will all survive to adulthood. But by producing lots and lots, they're gambling that enough will survive into adulthood. And crucially, there'll be variation occurring, so they'll be non-identical to each other. You can see here we've got some that are darker and some that are lighter. Second stage is this thing called selection, hence why it's called natural selection. It's called natural selection because nature is selecting which individual is best adapted. So by chance, it's just luck. Some individuals are better adapted than others just because of the genes they happen to have inherited, and they're more likely to survive. And then the final stage is inheritance, which is simply saying that those best adapted ones are more likely to survive and therefore pass on what we say are good genes, genes that increase the chance of survival. Or the opposite, the ones that are not well adapted are less likely to survive and will not pass on their genes. So the better ones become more common and the less good ones become less common, as long as the climate doesn't change. If the climate changes, then the good can become not so good and the bad can become very good. It's not a constant aiming towards the perfect deer, it's just what's best in that moment. Is it going to always be the best adapted? No, because climates change, environments change. And how can climate change affect natural selection? As I just said, climate change can affect which variation is best or fittest. Then next example is antibiotic resistance. Worth knowing a little bit about this because it's usually important, but also because it may well come to your exam. They quite like asking about this particular topic when they're doing natural selection questions. So antibiotics, we'll talk about in topic 5. They kill bacteria and they're amazing and we would be lost without them. They don't do anything to viruses, but they are phenomenally good at killing bacteria. And bacteria causes lots of problems. The reason why 100 to 150 years ago, lots of children died before the age of 2 or 3 was because of bacterial infections. We've basically got rid of that, but antibiotic resistance could move us back towards that. So it's something we really need to get right. Antibiotic resistance is what it sounds like. It's when the bacteria stops being affected by the antibiotic and can survive in the presence of the antibiotic. It's very bad news. So again, the reason it's bad news is it creates bacteria, super bacteria if you wish, and we can't treat them. They are free to do whatever they want to us or inside us. Complete the table to show how it occurs. So I've got a little key here. So blue are normal bacterium, yellow are resistant bacterium, and purpley funny shapes are dead bacteria. I should say it doesn't matter if you say bacterium or bacteria, it doesn't matter, it's single plural, but it makes no difference at GCSE. So what we have initially in stage 1 is a population of bacteria. This could be growing in your throat or in a wound or on a plate, it doesn't really matter. This one here, just by chance, is resistant to an antibiotic. It's not deliberately done that, it's not hoped for that, it's not changed, it's been born or produced and it is resistant because a mutation has occurred in its DNA. So let's say this is in your throat, you have a sore throat, you go to the doctor, they give you an antibiotic, and the antibiotic will kill all of the normal bacteria, and this one just hasn't died yet, but it will be dying. It might get lucky and survive, but it will more likely than not be exposed to the antibiotic and die. This one obviously is totally fine and unaffected. And what we've created here is a location where this resistant bacteria can grow very well, because it's in the throat where there's lots of resources. You can see there's lots of resources because the bacteria were doing great before the antibiotic. So it's going to suddenly explode in population. You can see that's exactly what's happening here. The blue ones are continuing to die. There's always a few kind of hanging on because of dominant recessive nature of genetics. But overwhelmingly, the population is going to become a resistant population. And then finally, worst case scenario, the entire population becomes resistant, and that antibiotic your doctor was giving you is totally useless. You may as well just take a deep breath. The air you're breathing in is just as useful. And that is a massive, massive issue, because now forevermore, this bacteria is going to be totally and utterly resistant to that antibiotic. That antibiotic is now effectively useless to treat this condition. And you'd have to be given another antibiotic, but there you run the risk of that antibiotic having the same problem. So why is this evolution? It's simply evolution because the bacteria are doing something at the end that they couldn't do at the start. So the species has changed. I'm not too worried about this one or this one. So this is the first one that's triple only. So if you're doing the combined, you don't need to worry about this. But the pentadactyl limb, penta means 5, like pentagon. It's just a limb with 5 digits. Your hand or your arm is a pentadactyl limb because it's got 5 digits or 5 fingers at the end. Some examples are humans, horses, cats, bats, and whales. You don't need to know these, but it's worth understanding that these are some examples. And we use them for a whole range of things like flying, walking, holding, moving, swimming. But it's all the same thing. Does the pentadactyl limb have the same function in each species? No. And crucially, how does it provide evidence for evolution? It's simply that there is a common ancestor out there. There is something that humans, horses, cats, bats, and whales, or mammals, are all evolved from, which is now long extinct, but it had a pentadactyl limb, and all of these adaptations, you can see there's a common ground in most of them. The horse, the digits have fused, but the commonality between these structures means that there must be a common ancestor, and we can use that to work out where we've come from in terms of evolutionary history and which species we're closer and further related to. This bit here I'm not too worried about. It's just showing how human beings compare to other species, but it's not something to know in the course. It's just a kind of teaching point. The kingdoms you do need to know about. So the 5 kingdoms are animal, plant, fungi, protist, prokaryote. Animals are things like tigers, bats. Their description is they're multicellular, which means they're made of more than one cell. There's no animal that's just a single cell. Their cells always have a nucleus, and they never have cell walls. That's kind of a wee bit topic 1 revision. Plants, separate category to animals. Don't worry too much about knowing any of the examples, but I'm sure you do know some examples of plants just by being alive. Again, they have to be multicellular. They have chloroplasts, they have a nucleus, and their cell wall is made of cellulose. Then we have fungi, things like mushrooms, yeast. They're mostly multicellular. They're not always multicellular, but overwhelmingly they are multicellular, like these mushrooms are made of lots and lots of cells. They always feed on dead matter or waste material, like poo or vomit. They have a nucleus, and their cell wall is not made of cellulose. You could be asked to describe a fungi, and that's what you need to say to get full marks. Protists are mostly unicellular, so single-celled creatures, sometimes not. They have a nucleus, some have cell walls, but they're never made of cellulose. An example would be Plasmodium, which for what it's worth is what causes malaria, which kills lots of people. Then prokaryotes, which I guess you may think of as bacteria, are unicellular, they have no nucleus. Examples E. coli, but you don't need to know these. These are just so you can go OIC. And that's how we used to define life. This is based purely on kind of observations of behavior and appearance and where we found them in the world. We've moved on to the domain system. So anything that references the kingdom system now is effectively obsolete. It does come up from time to time. We talk about the animal kingdom, the plant kingdom all the time, but they're not terms we use in modern zoology and ecology. We have moved on to the domains. So this system is basically gone, and this system is a new system. The reason we've done that is because prokaryote is actually a vastly varied group There are bacteria out there that are more different than, say, you and a crab are different, so it was worth giving them two separate groups. So we now have three domains. We have bacteria, which are unicellular, no nucleus, and they don't have any DNA in their genes, which is unused. So every piece of DNA in their genes is used for making protein or is used for function. Then we have archaea, which are kind of like bacteria, but are not. They tend to live deep underground and survive by breaking down chemicals. They don't do photosynthesis. They tend to live around deep sea vents, a kind of sort of extreme locations we tend to find these guys. They're always unicellular, they have no nucleus, and they do contain unused DNA in their genes. So some of their DNA in their genes is just there for no reason. We don't know why it's there, but it is. And then finally, all of these sections here have all been chucked into one group called eukaryota, which you should remember from eukaryotes in topic 1. They have a nucleus, and their DNA contains their genes contain unused DNA. So for you and I, each of your genes has these chunks of DNA that are not actually in any way functionally part of what the gene is doing. They're just there. So this question comes up all the time. Make sure you're aware of it. So why have we shifted from this group, these 5 kingdoms, these 3 domains? It's because we've compared the DNA of the species, and we found that actually these 4 have really similar DNA. This one here has really different DNA. So it's all to do with the genetic analysis of the species that's allowed us to go, actually, we don't need 5 kingdoms, we need 3 domains. Okay, topic 2 is human being evolution. We're worth considering on our own, since that's who we are. And you need to know a little bit about these historical species. So the oldest fossil we have that is something approaching a human being, maybe older, we just haven't found it yet, is a fossil we've called Ardi. They lived about 4.4 million years ago in Africa. Here's an image of what we think Ardi looked like. And in comparison to modern humans, he walked upright from time to time. He seemed to have very, very long arms relative to his body, kind of more like a monkey than what you consider to be a human being. And their big toes were pointing out of the way, which meant they could grip trees and branches really effectively, which is something we obviously have lost. I'm not saying it's a complete list, but these are the ones you need to know. Next, kind of noticeable one was someone called Lucy. She lived about 3.2 million years ago, again in Africa. Here's an artist's impression of what Lucy may have looked like. She seemed to be walking upright much more than Ardi, and her toes were just like our toes, but kind of curved, so they didn't point out the way, so much less of a tree-dwelling organism than Ardi was. And then finally, a whole bunch of fossils, which we call Leakey's fossils, but includes things like Homo erectus, Homo habilis, a few other bits and pieces. They existed around about 1.6 million years ago, but obviously a big range within that. Here's kind of what they look like. It is becoming more human-like. They definitely walked upright, and there's a whole range of things you can learn about them, but that's all we do at this stage. I'm not going to do the timeline, don't worry about that. One of the main ways in which we learn about human development is to look at stone tools and how they have changed over time. So over time, stone tools have obviously become much more complex compared to how Ardi and his species were using stone tools, how Homo erectus and how we were using stone tools has changed an awful lot. So if the stone tools are becoming more complex, that's just that the brain is getting bigger or more sophisticated, because designing these complex tools is not an easy thing. So as the stone tools get more complicated, we've assumed, and we're probably correct based on fossil evidence and just good science, that the brain is getting bigger, the brain is getting more complicated. So here's some examples of some species. You don't particularly know these ages, but it's just making you aware that the level of sophistication in the tool use has changed quite dramatically. So Homo habilis existed for 1 million years, which is an awfully long time, significantly longer than human beings have existed, and they were using basically stones which they'd broken, and they were using them to scrape the meat away from bones. Homo erectus, which seems to have wiped out Homo habilis, but never mind, they were using stone tools to hunt, to dig, to chop, so kind of more sophisticated stuff. Again, these guys are also extinct. Neanderthals, you may have heard of, they were using spears, they were using pointed tools, and then human beings or Homo sapiens, which is us. We've not been around for very long, but we've developed arrowheads, fish hooks, needles, and not including kind of modern things like power drills and screwdrivers. These are kind of historical stone tools as opposed to things that we now make in a factory. So how can we determine the age of stone tools? There's 3 ways. We can work out how deep in the ground they were found. So deeper in the ground means they were older. And we can work out how complex they are, because less complex means they're older, because they're simpler. You can see by this table that they were more simple here than they are here. And we can also carbon date, not the stone itself. You can't carbon date something if it's not been alive. So you can't carbon date stone, that would be pointless. But you can carbon date something that was alive that is attached to the stone tool, like some flesh that's fossilized or been found, or like a piece of wood that was used to attach to the stone. So these first were nice and obvious. The final one, just make sure you're saying organic material attached to the stone tool, not just, you can't just carbon date the tool. That's not correct. Okay, the last one, artificial selection. There's quite a lot of triple stuff in here. So what is selective breeding? So selective breeding or artificial selection, well, there's 2 ways we can do artificial selection. So we talked about natural selection, which is when nature picks who to select and who to allow to breed by you are fit, you can breed, or you are weak, you cannot breed, which seems brutal. Artificial selection is when human beings pick who to select, who to breed. So what is selective breeding? It's when we pick which members of the species breed together, normally for something we're looking for. Some examples of selective breeding, so we have the sheep here. They've been bred for thousands of years to become really woolly, which we use to make clothing. Potatoes, again, they've been selectively bred to initially make them less poisonous and then to increase their starch levels to make them more nutritious. And then finally, greyhounds have been bred for speed. These are not the only examples, far from it. Every cattle species, every domesticated species, every food species we use has been selectively bred. Why might they struggle in the wild? It's because they've become dependent on us for food or protection. So this is great, it's very woolly, but it would overheat in the wild. It would get eaten and hunted very quickly in the wild, for example. So most selectively bred species are totally dependent on us for survival. But we're also quite dependent on them for survival. The stage of selective breeding, if it comes up in a test, this is what you write. So you pick the ones with your desired traits, so the biggest muscles, the fastest, the best sense of smell, the most fur, the quickest growing. These can be plants and animals. You breed them together. You pick obviously the best male, the best female. You make them breed. You then collect their offspring. You grow them to adulthood. You pick the best of the offspring and you breed them together. And you just keep on going forever and ever and ever. There's no end to selective breeding. How we use selective breeding in food plants. We use it to increase the nutrient levels in plants. We've used to increase vitamins, minerals, starch quantities, how fast they grow. Kind of anything positive about any crop species has been produced by selective breeding. Domesticated animals, so we're thinking cattle, that would be how quickly they grow, how much meat they produce, how the meat tastes, how easy it is to harvest. We have selectively bred chickens that don't have feathers, for example, which means that they're easier to harvest and also they're more comfortable living in hot climates. Dogs have been bred for speed, size, smell. If you're a dog owner, I'm sure you're completely aware of this. Some disadvantages of selective breeding, it's far from perfect, is the first one is it takes a long time. You can't just breed 2 dogs and the offspring will be able to fly. It takes generation after generation after generation, so hundreds and hundreds of years. There's no guarantee it will work. Sometimes it works, sometimes it doesn't. There's a huge element of luck to selective breeding, but by doing it a lot, it becomes more likely it will work. It also produces offspring that are very similar to each other in terms of their genetics, which means they're very vulnerable to a new pathogen or a change in climate, for example. The second type of artificial selection is tissue culture. This is triple only. So tissue culture is basically when you get a plant, you chop it up into tiny pieces, and you grow each piece into a new plant. You couldn't do this with animals. If I take a human being and chop them up into tiny pieces, they die. I don't grow hundreds of people from each of those tiny pieces. Whereas a plant, you can effectively take one plant and make an infinitive number of plants, which would be genetically identical to that original plant. So why is it a good thing to do tissue culture or grow plants via tissue culture? You get lots of plants very, very quickly, and the plants will all be clones, so they'll all have the same characteristics. So if one makes really tasty fruit, they'll all make really tasty fruit. And if one takes precisely 12 weeks to be ready to harvest, they'll all take about that time to be ready to harvest. Okay, so hopefully you'll get a chance to do this. But how do you produce multiple plants from a single cauliflower plant? You simply take the cauliflower you're interested in, you chop it up into small pieces, and then you grow each one separately. You have to be very careful with infection and bacteria and giving them antibiotics and making sure they've got nice soil to grow in. But one cauliflower should be able to produce hundreds of thousands of genetically identical cauliflower plants, which if they have good traits like tasty or grow quickly, is a really good thing. How would these offspring compare to one another? So you can say they're clones, or you can say they're genetically identical. It wouldn't be right to just say they are identical, because they're not. Some might be slightly bigger, slightly smaller, might weigh more, might weigh less, but their DNA will be 100% identical. Here's our diagram showing the stages of plant tissue culture. So first thing is we take our plant, we cut off the piece we want, so it just needs to be a few cells big. We give them hormones called auxins, which we'll learn about in topic 6 in the future, and we basically grow them. So it's basically giving them all they could ever hope for and need. Each little sample becomes its own separate plant, and once they're big enough, we put them in soil, put them in compost, and great, we have a huge population of plants which are all clones of the original plant. So, a bit of problem solving here. Design an experiment to show the effects of different alcohol concentrations on liver cells. So you would take liver tissue samples. So you could take that from someone who's had the liver removed for some reason or has died and donated their body to science. Or you could just go and buy liver from Tesco. It wouldn't be a human being, but it works perfectly well. Place each sample in different concentrations of alcohol overnight or for an hour or for however long you want, as long as you're consistent. And then after that period of time, you would simply test the cells by measuring the catalase levels, for example. So that would be putting them in hydrogen peroxide, seeing how much bubbling there is. And obviously, the less active the liver, the greater the impact the alcohol has had. Obviously, the fact that the liver has been chopped up will also have an impact, but you can control that and you can mitigate for that. Why is it better to use tissue culture samples over transplant? It's because the tissue culture, so you grow these liver cells separately from a single liver rather than using multiple transplanted livers, is because we've got better things to do with transplant livers than test alcohol theories. So you want to give the transplant livers to patients and use the tissue culture livers for research. State some uses of animal tissue culture in medical research. It's basically this. You can fundamentally, all you're doing is growing populations of cells on plates. It's exactly this, but without this compost stage. You can use it to study how cells communicate. You can study viruses because a virus will need a cell to grow in. So you grow your cells, you infect them with viruses, and you see how they work. We can grow cancers or tumors in tissue culture and see how they go. There's lots and lots of things we can do. But these are the ones you should be listing in a test. Genetic engineering is the third and final way in which we can do artificial selection, and I guess it's the most famous one because it has a bit of controversy unfairly attached to it. So here's some examples. You don't need to know this, but the chicken is obviously not real. This is a Photoshop. This is a Photoshop. But I mean, this is real, but it's not GM. It's an ear that's been grown on the back of a mouse before being attached to a person. The mouse doesn't have amazing hearing. It's just there. This is a plant that grows in dark. This is the one example of genetically modified. So they've taken a jellyfish gene, they put it into the plant. Plant doesn't behave like a jellyfish, it doesn't want to swim underwater or sting people. It just has a gene which makes it glow. So, define genetic engineering. It's simply moving a gene or a piece of DNA from one species to another, always across a species. 3 examples of desirable characteristics that could be introduced. So anything positive is a correct answer here, but drought resistance, faster growth, higher nutrient levels, pesticide resistance, less fertilizer dependency, can grow in hotter temperatures, anything positive. As long as there's a gene out there which makes that happen, we can put into a plant or an animal and GM them, and it's a brilliant thing. The flowchart showing the key stage of genetic engineering. So First thing we do is we find the gene of interest. So for human beings, for example, one of the genes we have used in GM is insulin, to make insulin for diabetes or type 1 diabetics. Key thing here is you know the name of the enzymes. So we find the gene of interest, which we use the Human Genome Project for, which you've studied in topic 3. We find the gene and we cut it out using restriction enzymes. These are like scissors. They go in and they chop the DNA, but only at certain points, and they cut either side of the gene, and we have this isolated gene which we can then put in somewhere. At the same time, we take plasmids, which you learned in topic 1, are little rings of DNA that are found in bacteria cells only. We cut them open and we use exactly the same restriction enzyme, or same type. It might not be physically the same bit of enzyme, but it's the same type of enzyme. We then mix the gene and the plasmid together in a petri dish in very, very small quantities. And there's an enzyme which we add called ligase. Ligase is like a glue, and it will join the gene of interest to the plasmid, and you'll have these genetically modified bacteria once they take up the plasmid. And you can get the plasmid into the bacteria by injecting it in. You can fire it in with a gun. You can just mix them together, and the bacteria will happily collect the plasmids and bring them in themselves. There's lots of ways you can do that. Now you've got your GM bacteria which have this new gene. Every time they divide, they will continue to have the gene. They'll keep copying the gene because they copy all of their DNA, all of their plasmids every time they do mitosis, from topic 2. And you end up with a large population which are all genetically modified, and they all produce the product. So, for example, we now have huge vats in laboratories which contain effectively infinitive numbers of GM bacteria, and we have a tap at the bottom, and every now and then we open the tap and we get insulin coming out, which we then clean, sterilize, and then give to people who are diabetic, and it's the best treatment for them until we can use stem cells to actually cure type 1 diabetes. This is quite a difficult concept, but it's really important in terms of how restriction enzymes work. And it's the idea of a sticky end. So when DNA is cut by these restriction enzymes, it doesn't cut straight through like a vertical line. It creates kind of a jagged cut. And the reason for that is it allows DNA to be cut and then rejoined. If you're thinking, what's the point of a restriction enzyme? We find them in bacteria, and what they do is when a virus puts its DNA into a bacteria, they use these restriction enzymes to cut the viral DNA up before it can start doing anything bad. So they're actually a real thing, a natural thing, that human beings have just used for a new purpose. So we get the cut. This is the plasmid DNA, or this is the gene of interest, so it doesn't matter which one is which, but one's plasmid, one's gene of interest. They both get cut, they both leave the same overhang, and that means they can join together using good old topic-free base pairing. So here's TTAA, here's AATT, and they will bind via hydrogen bonds. So this one is restriction enzymes, it's cutting. This one is ligase because it's joining. And we now have a gene that's been inserted into a plasmid and is good to go. Why are sticky ends important? It's because the single-stranded overlap, which is this bit here and this bit here, allow the DNA to be joined together via hydrogen bonds and base pairing. If it was just a vertical cut, your body would, or any organism would really struggle to rejoin that. We don't really have the facilities to rejoin them that way because we don't know where they should go, particularly if you've got chromosomes. It's very hard to join double-stranded breaks back together, but if there's an overhang, you can just stick that back together using base pairing. Now, explain whether we use the same or different restriction enzymes for this process. We'd use the same ones because each restriction enzyme produces a different cut. So whatever this one is, there's one called BAM, for example, after the kid from the Flintstones. This one always creates a TTAA cut or an AATT cut. That means we can easily join them together. If the plasmid and the gene of interest were cut using different restriction enzymes, you'd have different cuts, different bases, and they wouldn't be able to pair together. They wouldn't be able to bind because T can't bind to C, for example. So why is that important? It's to create these complementary base pairs, like I just said. Explain the importance of vectors in genetic engineering. We kind of bounce around here. So a vector is simply a way to get DNA from one organism to another. In this situation, the plasmid is the vector. Give an example of a human product that's produced by genetic engineering. Insulin is the most famous example, but there are others out there like growth hormone. And then this bit's a bit repetitive, but let's quickly run through it. Advantages of genetic engineering is it can produce individuals with useful traits, which increase yield, decrease fertilizer or pesticide use, could grow in harsher climates, produce food that takes longer to spoil. That's great. The disadvantages are they're expensive. Some people think the genes might escape into the wild, and some think that GM plants can be bad for health. But there's no evidence of these things whatsoever. It's just hysteria. Give one advantage and one disadvantage of adding genes to crop plants. So it's not just a random gene, it's a gene which makes them resistant to insects. So the advantage is it prevents the insects eating the crops, which means you don't have to use as many pesticides, which protects the local environment and means you have more crops to sell to people and allow people to survive. The disadvantages are the same old stuff. So they are expensive, and some people are scared they're going to escape into the wild, but they won't. It's not a problem. Two other ways in which it can increase crop yield: anything sensible, growth rate, less fertilizer use, drought resistance, grow in harsher climates, anything sensible. Again, triple only, but these are some other ways in which we can increase yield. So we use fertilizers, which is what we kind of do right now. Fertilizers are simply putting chemicals into the soil which make the crops grow faster, stronger. The advantage is it gets a lot more yield. Disadvantage, you learn in topic 9, is that they can do all sorts of damage to the local ecosystem. Biological control is using a new species to control a pest species. Like if you have a cat and you want to get rid of it, you buy a dog and the dog eats the cat. It does deal with pest species, but the problem is that it creates a new problem. So in Australia, for example, they had big problems with insects, so they introduced cane toads. Cane toads did indeed eat all the insects, but now they have far bigger problems with cane toads because they're poisonous and they're killing other animals and they're kind of destroying ecosystems. So biological control is a nice idea, but it has to be very, very carefully controlled to make sure the thing you're introducing doesn't totally destroy the ecosystem. Summarize some of the potential benefits of genetic engineering and selective breeding in agriculture and medicine. So the benefits are it can produce individuals with useful traits, it can increase yield, it can decrease fertilizer pesticide dependency, they can grow in harsher climates, and they can produce food that takes longer to soil, which is all wonderful, wonderful things in a world where we're running out of farmland and we have a growing population. Some of the practical and ethical implications are that they are indeed expensive. Some people are worried the genes will escape into the wild, which probably isn't a huge issue because these genes are coming from the wild. Some people think GM crops are bad for your health when eaten. There is no evidence of that whatsoever. And in fact, most of the ones that we have evidence for, there's no difference to taste at all because we're not putting taste genes in, we're putting something else into the new species, or the new GM organism. Selective breeding's drawbacks are it takes a long time, it doesn't always work, there's no guarantee good traits will be passed on, you're kind of just rolling the dice and hoping that you get the number you want, but if you do it enough, you'll eventually get the number you want. And of course, it produces very similar organisms which may be vulnerable to changes in the climate. And that's that topic. I'm not going to go over the word bank, you can do that yourself, and I'm not going to go over these questions at the back again, you can do that yourself. Right. Thank you very much.